Sometimes when we're looking at a chemical reaction drawn out for us as substrates moving to products, sometimes it's hard for us to look at each molecule and know what's been oxidized and what's been reduced. One of the things that happens in a living cell that we can use to help us know where the oxidation is going on, where the reduction is going on, is the fact that for each of the electrons that get transferred or shifted in a bond, that we usually see an accompanying proton. In other words, the molecule or the atom that gains an electron or relatively gains an electron also tends to gain a hydrogen. The molecule that loses will also lose a hydrogen ion or a proton. Remember that when we use the term proton as it relates to molecules inside of a cell, we're talking about a hydrogen ion. So we follow this rule, which is follow the proton. If we go back to the reaction we looked at a minute ago, we can see that as this carbon atom sat in a carbon dioxide molecule, it was accompanied with two oxygen atoms. And we said that this carbon atom was going to be reduced during photosynthesis. It was going to experience a relative gain in electrons. It also got a proton. And when we looked at the water molecule, particularly the oxygen atom here, we said this was going to be oxidized during the reaction. You can see that it's associated with two hydrogen ions in the water molecule. But when it gets oxidized, those hydrogens are lost. So it sometimes helps when you're looking at a chemical reaction to follow the proton. And that will help you understand which atom, which molecule is going through oxidation and which through reduction. We're not going to be spending time in this class looking at chemical reactions and figuring out which molecule is getting oxidized and which is getting reduced. We're more interested in what's happening to the energy. And remember, we can follow the flow of energy by following the flow of electrons. The flow of energy in living cells is due to the movement of these electrons. And as we spoke of earlier in the lecture, one of the options for a cell in terms of what to do with free energy is converting that free energy into a form that is more helpful for the cell in that moment. It is the time that an electron or electrons spend in motion that the energy associated with those electrons is available to do work. So, energy is moving in a living cell, and the cell needs that energy to do work. Every time that bonds are broken and electrons move, there is an opportunity for a cell to use that free energy to do work. There's also an opportunity for the cell to store that energy as potential energy in new covalent bonds. If neither of those things happen, then the energy is going to be lost to the cell. It's not being destroyed, remember. It's not being destroyed. It's being lost. And it gets lost in the form of thermal energy or heat. One of the things about living cells with regards to energy is that they are not good at making use of thermal energy. Now, with the exception of the homeotherm needing to stay warm to maintain a body temperature, with the exception of that, living cells are just not good at managing thermal energy, making use of thermal energy, and so Thermal energy is typically lost. It escapes the cell and it is lost to the universe. This is part of the reason that cells age and eventually die. Remember, we talked about the issue of DNA replication and the telomeres being lost off of linear chromosomes with each round of replication or cell division. That too is part of the reason that cells have a limited ability to divide, the loss of their telomeres. But another reason that cells age is because during every chemical reaction that goes on in a living cell, a little bit of the energy that is in motion during that reaction is going to get lost as heat. So even when the cell does transform the energy or store the energy, a little portion of it will be lost as heat. This is part of the reason, again, that cells don't live forever. Let's move on now and start a discussion of measuring energy in cells. I've been using this term free energy. And if you remember from chemistry, when it comes to free energy, we think about the energy that is associated with a reaction that is available, that's available to do work. And we give this free energy a name. We call this Gibbs free energy, and we use the symbol capital G to describe it. Now, every molecule has a G associated with it. In other words, we know that every molecule, which is made up of atoms joined together with covalent bonds, we know there are electrons in those bonds, and we know those electrons have some energy associated with them. Energy is stored in a molecule. So all of that potential energy is measurable. Every molecule has its own G associated with it, its own potential energy associated with it. Now we express G in different ways, and we use different measurement systems to express it. It can be either be in the form of kilocalories per mole. Sometimes you'll see it as kilojoules per mole. But when we look at a reaction and we think about the G that's associated with each molecule in the reaction, we can also follow how the molecules change and how the G changes. So we can measure what we refer to as delta G, or the change in free energy that's associated with that reaction. Now, when you were in chemistry class, you learned a formula for delta G. We're not going to use that formula, though. We're going to use this formula because this makes a little more sense to us on a practical basis in a living cell. We're going to say that delta G is calculated by taking the G of the product molecules and subtracting the G of the reactant molecules. Products minus reactants. Now, why would we care about measuring delta G? Well, it turns out that the delta G number is going to tell us a little something about the reaction. When the delta G number comes out negative, it tells us that we're looking at an exergonic reaction. We're looking at a reaction that is going to release energy. In other words, a spontaneous reaction. In comparison, when we calculate delta G and it comes out positive, we are looking at an endergonic reaction, an energy consuming reaction, or a reaction that requires energy, and therefore a non-spontaneous reaction. Remember, when it comes to living things, the bulk of the reactions that are going on inside of the cells are non-spontaneous. They require a source of energy, they consume energy, and they also require an enzyme. Now, on this slide, we're looking at a graph that shows us the effect of adding an enzyme into a chemical reaction. On the x-axis in this graph, we can see just time going by. The reaction is progressing as we move from left to right. On the y-axis, this is the measure of free energy. So low amounts of free energy at the bottom of the y-axis, and more free energy as we go up. Now, this particular reaction involves a molecule called A being combined with a molecule called BC in order to create two new molecules, two product molecules, one of which is AB and the other one is C. All right, so essentially during this reaction, the covalent bond between BC is going to get broken, and a new bond is going to be built between A and B. Now, every molecule has its own G associated with it, so A and BC have their own G associated with them, and together that brings the reactants to a free energy level right here, where the red line hits the y-axis. That's the free energy level at the start of the reaction. And if we follow this line, and in this case we're going to go up the dotted line, we can see that free energy starts to rise and hits a high peak. This is the transition state of the reaction. And if you can recall from your earlier coursework, we defined this state as sort of the in-between state of a reaction when all of the substrate or reactant molecules, all of those bonds have been broken down, but none of the new bonds have been built yet. It's a transition state. All of the old bonds have been broken, none of the new bonds have been built yet. We're at a high level of free energy because all those bonds have been broken down. So we're at a much higher level. If we go from the peak over to the y-axis, we've gone way up in free energy. Now, that free energy is going to drop as new bonds are made, and we ultimately end up with our products. And if you trace this red line over to the y-axis, you can see that the products have a lower level of free energy than the reactants did. In order to calculate delta G, we take the G of the products, this lower number, and we subtract the G of the reactants, this higher number. So for this reaction, delta G is negative. That tells us something. That tells us that this is a reaction that is exergonic and likely spontaneous. Now, what exactly does that mean? Well, think about it this way. If we started off with reactants that had this level of free energy, and we ended up with products that have this level of free energy, we've had a drop in free energy between the reactants and the products. That energy had to have gone somewhere. You can't create energy and you can't destroy energy. So if your reactants are up here and your products are down here, that means some of the energy in the reactants had to have been released. It's not in the product molecules. It's not stored in the product molecules. It had to have been released. That's what an exergonic reaction is, a reaction that has a release of energy. Now, let's take a look at what happens when you add an enzyme into this reaction, because as I said a minute ago, most biological reactions require an enzyme. We're going to start off with the same reactant molecules, and we're going to end up with the same product molecules. But this time, as we follow the progress of the reaction, we're not going to travel up the dotted line. We're going to continue on with the straight line. And we're going to see a rise in free energy as bonds are broken, but our peak stops right here. It then comes back down and we build new bonds to make our products. This is the effect of an enzyme. What an enzyme does is it lowers the energy needed to get to the transition state. It lowers what we call the activation energy, the EA. It lowers the activation energy, which is the energy required to hit that transition state. It's easier, in other words, to hit the transition state when you have an enzyme. And that's because the enzyme is doing something important. It's bringing the substrate molecules together. It's making the reaction easier. It's allowing it to happen more readily. It's reducing the EA, the activation energy. And that means you're going to get your product formed at a faster rate. Hopefully that is review for you as I talk about enzymes. But today I want you to notice something else on this graph. When we added the enzyme, we lowered the EA, we lowered the activation energy, the energy needed to hit the transition state. But you know what we didn't do? We didn't change the delta G at all. The energy associated with the products and the energy associated with the reactants, that didn't change. The delta G, the G of the products minus the G of the reactants, does not change. It is inherent to the reaction, which is why we can name a reaction an exergonic reaction. Name it as being endergonic. It doesn't matter whether or not an enzyme is involved. The nature of G doesn't change. Now, why is that important? Well, it tells us that exergonic reactions are always going to be exergonic reactions. Endergonic reactions are always going to be endergonic reactions, and both kinds are going on in living cells all the time. Exergonic reactions are releasing energy. Endergonic reactions are consuming energy. And in a living cell, we don't want to let any of that energy go to waste. So, what does a cell do? Now, it would make sense for a living cell to bring those two reactions together, to couple the exergonic reactions and the endergonic reactions so that the energy that gets released by an exergonic reaction can be used by the endergonic reaction. That makes lots of sense, doesn't it? We're going to couple those two reactions together, and we're going to let one reaction provide the energy for the other reaction. But here's the problem. Those reactions don't necessarily occur at the same time. So it's not easy for a cell to bring those two reactions together in real time. A cell can still couple them, though, by using a middleman molecule. A middleman molecule that can capture the energy released by exergonic reactions and hold on to it temporarily and then release it in time for the endergonic reaction to occur. We need that middleman. And that middleman, of course, is ATP. ATP is often described as the currency of energy. Just like actual currency, dollars and cents, couple someone who's trying to sell an item and someone who's trying to buy an item. So the ATP couples reactions that are releasing energy and reactions that are consuming energy. And it gets rid of the problem of those two things happening at different times in the cell. So, how does this energetic coupling work? Well, this is sort of the classic diagram. Remember, the energy is being carried with electrons. So, when exergonic reactions occur, an energy is released. what the cell can do is capture those electrons in covalent bonds. And specifically, it will be a phosphate bond inside of this ATP molecule. So as energy is released from exergonic reactions, the cell will take an ADP and build a covalent bond to a phosphate group. And that's going to create ATP. So all of the energy that gets released from these exergonic reactions is going to be temporarily stored in that phosphate bond, in those ATP molecules. Now that ATP will sit in the cell until this happens, until endergonic reactions need to occur and need a source of energy. Now the cell can break that phosphate bond in the ATP molecules, take off that phosphate from ATP and create ADP, and release the energy that comes from those electrons and fuel or supply these reactions. This is a description of energetic coupling. This is how ATP is used by a cell. Now that's an important point here, because when we talk about the cell taking ADP and phosphate and building ATP, it's tempting to say the cell is making ATP here. I mean, it is literally making ATP, but I want you to think about it as using the ATP, because this is not how a cell makes up a supply of ATP. It does that through cellular respiration. It makes its ATP through cellular respiration, but it uses its ATP in energetic coupling. And it uses it by breaking it apart right here, and then building it back up, and then breaking it apart, and then building it back up, and breaking it apart, and building it back up. Releasing energy, storing energy. Releasing energy, storing energy. This is how cells use the ATP that they make during cellular respiration. Now, when we talk about energetic coupling, we can see it occurring in lots of different reactions inside of cells, because endergonic and exergonic reactions are going on all the time in living cells. Let's take a look at this reaction. C6H12O6, this is glucose, combining with oxygen to produce carbon dioxide, water, and energy. Do you recognize this reaction? This is cellular respiration, and oxidative phosphorylation would follow. So cellular respiration is glucose being oxidized all the way to carbon dioxide, and all the energy that gets released from the glucose will be used to make ATP. If you were to take that glucose and put it into a test tube and set it on fire, you would also oxidize the glucose. You would break the covalent bonds and release the electrons. But of course, if you did that, all of that potential energy would be lost. It would be lost in the form of thermal energy or heat. You would have transformed the potential energy into thermal energy by doing that. And you would lose that energy out into the universe from your test tube. Inside of cells, we want to prevent that loss as much as possible. So inside of the cell, as the glucose gets oxidized, first of all, it's going to get oxidized very slowly. The glucose is not directly converted to carbon dioxide. The covalent bonds are broken one at a time through a long series of redox reactions. Remember, during respiration, cellular respiration, the glucose is first broken down into pyruvate. The pyruvate is then broken down into acetyl-CoA and carbon dioxide. And then the acetyl-CoA is broken down into carbon dioxide. Glucose to pyruvate is what we call glycolysis. Pyruvate to acetyl-CoA is what we call pyruvate processing. And acetyl-CoA to carbon dioxide is what we call the Krebs cycle. It's a long series of reactions to oxidize glucose in a living cell. Along the way, as all those bonds are being broken, electrons are being released. The cell does not want to lose that energy as heat. So it's captured. The electrons are captured, and they're then used to do work. And the work is make ATP. By building the ATP, they now have a little molecule, lots of it, that has a nice little bundle of energy stored in its phosphate bonds. And it can turn around and break those phosphate bonds in order to release that energy and do more work. In 1 mole of glucose, there are 686 kilocalories available to the cell. The cell is going to break apart the glucose molecule, release that energy, and then harness or store it by making ATP.